these are 3 papers published with Alessia. And, you know, they go from 2019 to the last one, 2026. So I will put together 2019 and 2026. And as you notice, I'm very proud of her work because we managed to simulate the whole chromatophore. I don't know if there is a laser pointer. Yes. The whole chromatophore with more than 5,000, almost 5,000 chlorophyll bees, bacterial chlorophyll molecules. And the whole chlorosome in green sulfur bacteria that contains more than 100,000 molecules, okay? So thanks to our work, we were able to do these huge, large-scale simulations. But of course, what I want to tell you today is what is the deep meaning of our results, okay? What do all these simulations and work tell us of interesting? I will find, let's say, the main message, or one of the main messages. So again, this is about to keep the theme of the conference, quantum in biology, and then biology for quantum is our Horizon project that I will mention if I have time at the end. So let's start. As you know, biological systems, many biological systems are characterized by high degree of symmetry. You know, chlorophyll molecules self-aggregate in huge cylindrical aggregates in green sulfur bacteria. There are 4 concentric cylinders. Here only 2 are shown. And they form the chlorosome. And then there are the FMO below that transmit excitation to the reaction center. But also, like Philip Kurian and Travis Craddock showed, and this is the work I did with them, we consider microtubules that can form beautiful structure like the centrosome and so on. I will focus in this talk only on the green sulfur bacteria. But again, you can find more details in Alessia poster. So the question we want to ask is, how is it possible that in large aggregates we can have coherence at room temperature, okay? I mean, if we have a small aggregates and you have strong coupling and, you know, and the coupling is stronger than KT, then you have an excitonic state which spreads over 2, 3 molecules. That's fine, right? But if you have a large molecular aggregates, then within KT, you have many levels. And if you have many levels, they scramble, they mix with each other, and the coherence goes down. So how we can help to preserve how and if, let's say, biological system can preserve coherence at room temperature when they're big, okay? That's the question I would like to answer. And this is a famous calculation of the coherence time, the space. If you look at space, degrees of freedom, the coherence time at room temperature is very fast. But coherence time depends on the degrees of freedom you look at, okay? Indeed, superconductivity, ITC superconductivity, it's almost at very high temperature, and this is a quantum coherent effect. So it doesn't, room temperature doesn't mean you can destroy all coherences, okay? Now, what can you do with coherences, or what coherences do if they become a cooperative effect? Then coherences can introduce a fast time scale that can beat the phasing time scale, like super transfer. Or, as I will show you in this talk, coherences and cooperative effects can suppress the density of states close to KT, allowing to coherence to survive. Okay, and this is the precise, and what I will show you today is the precise mechanism that we found in which this happens in natural systems, okay? So first methodological, very simple methodological, let's say, parenthesis. To model this photosynthetic system, we use the radiative non-Hermitian Hamiltonian. You know, for the students here, when the light comes, the electron goes up and down in a classical picture. It's driven by the electromagnetic field of the light. So it's an antenna, right? It's not a static dipole. So this antenna will emit electromagnetic waves and it will excite only a bi-antenna of the other chlorophyll molecules. So this Hamiltonian, if you never saw it, then open the Jackson book of electromagnetism, and this is the antenna-antenna coupling. So that's what we're using. Why we're using these? Because in the limit of small volume, so when the wavelength is larger than the system size, then this reduces to the Frenkel Hamiltonian, dipole-dipole. But this non-Hermitian Hamiltonian allows us to study large aggregates. Like for microtubules, the lambda is 280, but the microtubule is microns long. You cannot use the Frenkel Hamiltonian, okay? So this is the only methodological part. Let's say I will try to stay general. Now, in order to show you what is the mechanism in natural system that allows coherences to survive at room temperature, with Alessia, we did this work. So we consider, nanotubes, different nanotubes of green sulfur bacteria. You have the mutant type. The mutant type is a mutation of the green sulfur bacteria. Is made of stacked rings. Each ring contains 60 chlorophyll molecules, okay? Then there is the wild type, the WT. It's really, maybe I go down because it's really very hard to look up there. Okay. The WT, the wild type, okay, this is much better. This is the natural, more natural. It comes in variation, but there are spirals, okay? Then we compare these to the natural models with mathematical models. In the parallel dipole model, we put all the dipole parallel, okay? Then here we put all the dipole tangential. So we take the same, let's say, position of the mutant type, but we change the dipole. We also consider randomly distributed dipole. And we also consider with and without long-range interaction. So we also cut and consider only nearest neighbor. Sorry, then we use our non-Hermitian Hamiltonian, and we see that, the mutant type has a superradiant state close to the ground state, as well as the WT. Basically, all the ordered system, parallel dipole, tangential dipole, they have a superradiant state. So they're all superradiant. The superradiance increases. So the dipole strength of the eigenstate with the most dipole strength increases with the length of the nanotube, apart from the random dipole, in which you don't have this cooperative increase, okay? Now, we study the coherence length. How do we study the coherence length? We take a thermal density matrix, so we dialyze our system, we take a thermal density matrix, and we consider the thermal coherence length. So this quantity, to make an example, if the excitation is only on one molecule, then this is 1 over n, where n is the overall number of molecules. If an excitation is mixed, equal probably spread, but incoherently over all the molecules, this quantity is 1. If it is coherently spread, this quantity is n, okay? And now we plot, as a function of the nanotube length, the coherence length. So you see the wild type and the mutant type are over here, almost 1,000 molecules, let's say 500, 600 molecules of coherence at room temperature, while all the other models are an order of magnitude or more below, okay? Now we wonder, okay, why this coherence length is larger in this natural system than the other mathematical models? All the couplings between these models, the nearest neighbor coupling are the same. Here I also show the nearest neighbor model, which is this one. So the nearest neighbor coupling are the same. Long range is present both in natural and in the parallel dipole or the tangential dipole model, in the random model too. So these models have all cooperative effects, and they have all the same nearest neighbor coupling and all the same long range. Nevertheless, natural model have a larger coherence, okay? Now, let's look at the density of states within KT. As you can see, the density of states is highly suppressed in the natural model with respect to the other models. And now, what I want to tell you, this is what I already told you, the same coupling, the same long range, cooperative effects, but natural model have 1 order of magnitude more coherence than the other mathematical models. So, and the precise mechanism that, thanks to Alessia and Francesco Mattiotti, another brilliant ex-student of mine, we were able to identify is the following. So, this is a single ring, right? You remember the mutant type model is made of rings, stacked rings, each with 60 molecules, okay? Now, this is the dipole strength of one ring. You see the mutant type have 3 superradiant states. This is the ground state, first excited state, second excited state. The tangential dipole is also 2 superradiant states in its ring. The parallel dipole has 1 superradiant state, but at very high energy, okay? Now, because of the symmetry of these cylinder, each eigenstate couples only to the corresponding eigenstates in the other ring. Okay, this you can prove mathematically, and we did prove mathematically, okay? It's not difficult. So here I show you, we take 2 rings and we pull them apart. And we compute in red the coupling between the superradiant states. And then in blue is the coupling between subradiant states, maybe state 14, for instance, with the corresponding state 14 of the other ring. And in red, sorry, in green, in black was the superradiant state. And in red, there is the strongest coupling between singular molecules, okay? So as you can see, these superradiant states in each ring couple strongly between each other. And even the distance, the K of the coupling is not 1/r at the 3, like you would expect for a dipole, but it's 1/r at the 2 at first, compared to 1/r at the 3 of a single molecule. And subradiant states even faster, 1/r at the 5, okay? So basically what happens in this ring, I conclude the mechanism is the following. In natural system, this happens, so all these models have superradiant states that couple by supertransfer, okay? But in natural system, the superradiant states in each ring are the ones that produce the lower part of the spectrum. So since the lower part of the spectrum is produced by eigenstates which are strongly coupled by supertransfer, then of course you have a stronger coupling, you have a suppressed density of states. Okay, so the way natural system, and even you have the emergence of an energy gap. This is an energy gap as a function of the length that emerges in, is not so much, it's 10 centimeters to -1, but this is amazing if you think that I'm increasing the system size and the distance between the ground state and the first excited state increases instead of decreasing. Okay, yeah, I'm changing the number of molecules from few hundred to 3,000. And this is not the number of molecules, this is Armstrong. So I'm going up to 200 nanometers. And by the way, 200 nanometers is the natural length of this system. So this energy gap increases up to it reaches a peak around the natural length of this system, okay? So as you can see, I was very proud of this work because it's a precise mechanism in which you can preserve coherencies by suppressing the density of states using cooperative effects in large molecular nanotube aggregate. Then in this work, always with Alessia, we study the energy transfer of these Green-sulfobacteria, okay? So we take 3 concentric cylinders. These are, each of these cylinders is made of 4 concentric walls. We put 3 of them. We have 132,000, right, Alessia, molecules in this system. Then we simulate sunlight with a black body spectrum that takes into account the distance Earth-Sun. So we depress the density of photons due to the distance. So we take into account the solar spectrum. We compute the absorption. Then we take a dimeric base plate, which is the, let's say, most reasonable model for the base plate in Green-sulfobacteria. This is the position of the dipole. These are also chlorophyll molecules in the dimeric base plate. And then with a master equation, we study the energy transfer. We use MC fret between, technically, we use MC fret between the cylinders. And we do many methods, but basically MC fret between the cylinders and the base plate. Then there is, we don't model the FMO and the reaction center, but we model them as a trapping rate present on the bacteria, on the base plate, which is based on the actual real density of these FMO RC complexes on the natural base plate. Okay, and then we compute the current as a function of the trapping rate. And this is the trapped current. So there are a certain number of photons that arrive on the surface, a certain number of photons which are absorbed, and we compute the actual number by taking into account the absorption spectrum of these structures, okay? Out of these photons, there are a certain number of charge separation events, and so you produce a current, okay? And this you can see how much current you produce as a function of this trapping rate. And what is interesting that this is the natural range of trapping rates that you have in natural systems. And this is the RC closure time. What is the RC closure time? You know, when the RC gets an excitation, it goes in a charge separated state. It cannot receive another excitation for a certain time until it gets reduced again, okay? This time can vary a lot in natural situation. Let's say from 100 microsecond to 1 millisecond, more or less, okay? So as you can see, this system operates in a regime where let's say, the natural trapping rate is within the RC closure time. This means the following, like if an excitation arrives more frequently, right, the RC is not able to process it. If an excitation arrives less frequently, then you could process more, so you're wasting energy, right? So this is kind of an optimal situation, right, for a natural system to be, in which the number of excitation that arrives is the same of the number you can process, okay? And this is the efficiency which reproduces in this window, the 80% or so natural efficiency. But now, again, we want to compare these results with other mathematical models. In order to do this, we take the mutant type and we change the dipole orientation. So this beta is 55 degrees in mutant type. This is the trap current. If we put beta 0, then we have the parallel dipole, okay? If we put beta 90 degrees, then we have the tangential dipole moment, okay? So as a function of beta, Alessia took a cylinder and put a dimeric base plate below it and computed the trap current as a function of beta under sunlight, okay? As you can see, for the parallel dipole, the trap current is this value, then it increases, it reaches a maximum, and we didn't do it by purpose, more or less around the 55 degrees of the mutant type. Then at 90 degrees, it reaches this value, it goes up again. Of course, it's periodic, right? And so on. So interestingly, the mutant type, so the natural structure, is the best in transferring the energy to this dimeric base plate. Now, before I told you that natural structure has this huge, let's say, coherence length at room temperature, but to conclude that is the coherence length that induces this huge, this optimal or efficient energy transfer is not so immediate. Because we tried, with Alessia, we discussed also today, how can we prove that is because of the coherence length, right? You can say, okay, maybe you add disorder, you decrease the coherence length. But when you add disorder, you also change the energy mismatch between the dimeric base plate and the nanotube. So disorder can produce different effects. Then you could start with a certain state which is localized and a state which is delocalized. But again, if they have different energy, then you don't know whether it's the localization or the energy. So I think that the fact that these natural structures are delocalized, we checked. They also, if we plot the MC fret, it has the same behavior, okay? I think that the localization plays a role, but we didn't nail it down yet, okay? That's because it's very complicated, because you have to change the localization without changing the other parameters, okay? But anyway, the general message I want to give you is that, let's say, cooperativity and the precise geometrical disposition can have a huge impact on controlling the coherence of the system, okay? And what I'm very happy is that we unveil the precise mechanism in which this geometry can suppress the energy density in this nanotube, enhancing the coherence length, okay? And finally, I want to mention this quantum biology for quantum. This is the Apache project in which we want to build a sunlight pumped laser able to operate on the one sun using this marvelous photosynthetic machinery. And finally, let me mention Nativa Calavera, which is the team of designers that are helping us in the project, okay? Young team of guys, very good, and so I'm done.
Thank you so much for the lovely presentation. Questions?
So I'm not very familiar with that, but I heard you say cooperativity. So that sounds quite novel in that you bring a new concept into the effect, the advantage of coherence. You add cooperativity. But from what I know, cooperativity is non-linear. So are you saying that some sort of non-linearity can enhance, amplify the effect of coherence?
Look, in the case of a single excitation, cooperativity is linear in the sense that scales that, if you have two molecules that coupled with V, and then you have N molecules with a giant dipole, which is square root of NV, so the coupling becomes square root N times V. So it's proportional to the number of molecules in each aggregate. This is the sense I'm talking about cooperativity. So here, you create this cooperative giant dipole in every ring, and they couple very strongly with the correspondent. Because of the geometry, they couple only with the correspondent eigenstate, and so they couple strongly because they're both giant dipoles. The strong coupling suppresses the energy. If the strong coupling is the one producing the eigenstates of the system, which lies below KT, then this produces a suppression of the density. And this is what happens in natural system.